Hey, this is Miss Hunt, and this is the notes for Chapter 9.2. We went over this on Wednesday, so I want to make sure I get a video up for you guys. Hope everybody's doing well. My online kids, I sent out a reminder yesterday, and I put something on our Google Classroom. We will not be doing online learning second semester. So if you want to continue to stay at home, you're going to have to switch over to Edgenuity, which means you're out of the honors classes. You still have a choice to take biology, regular biology, second semester from Edgenuity, but it won't be with me at all. So I highly encourage you to come back and join our class second semester, come back in January, but just make sure you have a plan of what you're going to do. So if you want to stay with me and stay in the honors classes, which I recommend, start coming back on January. Of course, you can come back earlier if you want to, but January, you need to be making plans to come back. If you want to stay online, you're going to be switched to Edgenuity, and there's no honors classes, and that's going to be handled by the Whitfield County Online Learning Academy, so it's a different teacher and everything. So hopefully I will see you soon. Okay, here is our Section 2 notes. Let me view this. Okay. So we have been learning the phases of mitosis, and we've been talking about them in class. We did some drawings. I hope that the drawings I did with you help you out. This is the notes for Section 2, and after this, you need to do the study guide for Section 2 for Chapter 9. Okay, we have the phases of mitosis. We said PMAT. PMAT is mitosis. Mitosis is PMAT. So the P stands for prophase. In this, the cell's chromatin is tightening up, and the sister chromatids are coming together and attaching at the centromere, and the spindle fibers are forming in the cytoplasm. So let's take a look. Here is the centrioles at the poles, and the spindle fibers are starting to come out. Those are the things that look like spaghetti noodles. All right, these are sister chromatids because they've doubled in the S phase, so each of these chromosomes are 2 chromatids, and they're held together by a centromere. I hope the amoeba sister video really clarified the difference between a sister chromatid and a chromosome and a centromere. All right, so in prophase, the nucleus is disappearing. You can see that little dotted line here. The nucleus is disappearing. The sister chromatids are coming together. So in prophase, we see a lot of phases of prophase. This right here kind of gives you the example of the phases of prophase. So we go from having a nucleus to seeing a centriole, and these chromosomes are condensing. The chromatin tightens up into the chromosome, and they're condensing. So I say it looks like a bowl full of noodles. The sister chromatids begin to find each other, and they attach the sister chromatids, and the spindle fibers start to form. And then we see that the nucleus is gone, and the sister chromatids are out. So all of this is prophase. So when you're looking at your slides, you're looking to see if you see a bigger than average nucleus and that it's starting to disappear, or you start to see what I see as a bowl full of noodles. Okay, let's look at this picture right here. This is in prophase. This one's in prophase. This one's in prophase. Right, these right here are in interphase. They're just being cells. They're in interphase. That's being a normal nucleus. But here you see that it's fading, and we're starting to see chromosomes condensing. That's what I would consider to be prophase. So when we're going through here, we're going to be counting for our lab, interphase, 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 prophase, interphase. Okay, possibly beginnings of prophase. You're going to see some of these that don't have any nucleus at all, and that could be that the cell is actually dead and it's just left behind the cell wall. So you don't count those as a phase of mitosis. You can just say they're just a cell. All right, what about, we'll come back to this one in just a minute, okay? All right, let's continue on. The nuclear envelope seems to disappear. That's the membrane around the nucleus, and spindle fibers attach to the sister chromatids. Okay, all right, the next phase is mitosis. So that's what it looks like, that the sister chromatids are being moved around until they get to the middle of the cell. So everything from here before is considered prophase of mitosis. All right, in metaphase, the sister chromatids are pulled along the spindle apparatus towards the center of the cell. So here's my sister chromatids. Remember, it's 2 chromatin, 1 chromosome, is being pulled into the middle. Okay. Sometimes they'll show this picture sideways, and they'll have centrioles in the north and south poles, and the sister chromatids are lining up in the middle, kind of like a belt. Sometimes they refer to this as the equator, that they're lining up at the equator. Equator is just meaning middle. So expect on an EOC that they're going to throw at you weird words, like instead of using middle, they'll use equator, just because they try to confuse kids on that. They line up in the middle of the cell. Okay, metaphase is one of the easiest to recognize. Let's go back and look at metaphase here. All right, right here, these cells are in metaphase. You don't see the perfect X's, no. What I think it looks like is stitches, or some people have said it looks like on a football, that the stitching on a football. Okay, that's what we would consider metaphase. All right, anaphase, the microtubules of the spindle fiber apparatus begin to shorten. So these little guys right here, the spindle fibers, are shortening up, and they're pulling the chromosomes apart. The sister chromatids separate, and the chromosomes move towards the poles in anaphase. We say anaphase away, and they're being pulled apart. Okay, let's take a look at it up here. All right, this right here would be an anaphase, and this one too could be an anaphase. They're being pulled apart right here. Okay, that's a more realistic picture of anaphase. All right, in telophase, the chromosomes arrive at the poles and begin to relax. We start to see 2 nuclei forming. If it's an animal cell, we start pinching apart what happened here, and we'll see 2 nuclei forming in the telophase. All right, finally we have cytokinesis. The animal cells are going to pinch apart and move apart. In plant cells, we start to see a cell wall. So let's see if we can find one on here. I'm not sure right this second. Let's see. Right here, this is this is a cell plate that's forming between these guys. That would be telophase, and that would be them pulling apart. Right here, this kind of looks like a cell plate coming up between 2 of them here. So that could also be a telophase. They pinch apart, or not pinch apart because they're plant cells. They're going to form a cell plate for cytokinesis. Okay. All right, here's another one that I left up on the board. We looked at microscope slides, but if you look at the mitosis reference materials, you should be able to see a lot of these pictures, so you don't have to use the microscope to see it. Okay, so that gives you a better look. There's prophase. These are just being interphase, normal cells. Anaphase away, telophase, 2 nuclei in the same square, and metaphase lining up in the middle. Okay, so after you go through these notes, you need to do the study guide for 9-2. And then you're also doing the lab today. So I just put this up today for the mitosis lab. So on the lab, we're going to go into this document right here. Okay, so this is a picture of an onion root, and you need to count how many cells are in each of the phases. Okay, so you definitely see a lot of interphase, but you have to count when they're in a different phase. Okay, you can use the text read write PDF reader to like strike through and highlight them to do a count on the side. That's what we're going to be doing in class. And this is a whitefish blastula. Basically, when an egg and sperm come together, they make one cell, and that one cell divides many times, like 100 times, before they become specialized. And then after that, that 100 cells, that's called the blastula. And then that 100 cells start to differentiate. Some decide to be the head, some decide to be a heart, some decide to be a spine, and we get differentiation. But right here, they're just the blastula. All right, so you will read the lab. You're going to make a table. How many is in each phase, and then do a percentage. So you do your total, and then how many of interphase out of the total, and you should get your percentage. You do that with the onion and the blastula, and then you're going to do your CER. Okay, do plants and animal cells spend approximately the same amount of time in each of the stages of the cell cycle? So you should be able to, based on your data, come up with this. All right, and that'll be our lab that we're doing today. Make sure you do turn this document in. If your PDF reader doesn't work, then you need to find a different solution. You can write your answers on a Google Doc. You can write your answers on paper. If it's on a Google Doc, turn that in or share it with me. If you do it on paper, you can take a picture of it and share it with me, or upload it on a Google Doc and turn that in. All right, good luck. Have a great day.